Hi, I'm Jim Phelps. If you don't know who I am, you could go to the trailer for this psychoeducation YouTube channel. Um, but let's get underway here. Thank you for coming. This is the video about preventing Alzheimer's dementia, possibly with tiny doses of lithium. This is a translation of an article that just came out in the journal Nature, which I think changes everything and changes a lot in psychiatry. The reason to look through this whole video is because if you're thinking about using lithium for the prevention of dementia, you really have to believe that this is the right idea and worth doing for a long time because you won't experience any short-term benefit. It's all about the long-term preventive benefit. So you really have to believe that this is worth doing. Second reason to look through this video is because the science in this Nature article is just astoundingly good. So a beautiful example of what our field can do. And psychiatry, in fact, looks pretty good here. And I think this is going to change psychiatry as well. So there are three main findings from this article. The first is that lithium in the brain, which is there naturally, helps prevent Alzheimer's. And the second is that a tiny dose of lithium per day could help prevent dementia. And thirdly, that lithium orotate, a particular kind of lithium, not the one we usually use in psychiatry, could be better for this than the carbonate version we usually use. A quick review of Alzheimer's dementia is caused by neurofibrillary tangles that interfere with the cellular survival and amyloid plaques that are also deleterious to neurons. So we're talking in this video about plaques and phosphorylated tau, which is the protein that contributes to these neurofibrillary tangles, ultimately resulting in neuron shrinkage and death. Here's an outline of what I'll be presenting in this video, starting with lithium in the brain, then looking at lithium deficiency, a dietary deficiency in mice that increases plaque and phosphorylated tau. Then some associated results from doing that lithium deficiency experimental work. And then the key to the whole thing here is, can lithium prevent or even reverse the Alzheimer's process? And then if one was thinking about using it, what are the risks of lithium in this orotate form? And then finally, what dose should one consider using? So starting here then with natural lithium in the brain. So these results are from doing brain autopsies on people who have died from various causes and people who had no cognitive impairment or minimal cognitive impairment, so-called MCI, or Alzheimer's dementia. And what one sees, and these are the data from that Nature article, I'm going to show you the uncut, unaltered graphs. But if you're not familiar with navigating this kind of graph, all you have to do is really see the main trend, and it's going to be obvious in every one of these slides. So you don't have to figure out what the axes are or what exactly all these other numbers mean if they're not familiar to you. What you see here, for example, is that comparing normal controls to people with MCI, minimal cognitive impairment, or Alzheimer's, the amount of available lithium in the brain, that cortical lithium, decreases across those different groups. The researchers were so stunned by this finding, they did it again in a different sample. They replicated it because they wanted to make sure they had this right. And they saw it again, that decrease in cortical lithium in people with Alzheimer's. So where did the lithium go? As we'll see, the implication here is that it went into the plaques, those A beta, the amyloid plaques. So for example, non-plaque lithium, lithium that's not stuck in those plaques, there was only half as much, 50% less in people with Alzheimer's than in the control brains. In minimal cognitive impairment, there was twice as much lithium in the plaque as there was in the non-plaque, and in Alzheimer's, 3.5 times more lithium in plaque than there was in the non-plaque setting, lithium that was not in a plaque. In addition, they looked at some statistics on cognitive impairment and lithium and measurement of non-plaque lithium, and found that the lower the non-plaque lithium, the lower the cognitive function scores. This is in humans generally. If you look at the entire aging population across lots of brain samples, if you're a statistician or familiar with these, you'll recognize that these are strong numbers, especially in the Alzheimer's group. The lower the non-plaque lithium, the lower the cognitive function. So our conclusion in this first segment then is that lithium appears to be sequestered in plaques in Alzheimer's, and it removed then from that non-plaque lithium space, and that lower non-plaque lithium is associated with lower cognitive function. Now we'll look at the experiments that were done by the team that published this Nature article, creating lithium deficiency, and what we'll see is an increase in plaque and phosphorylated tau. This lithium deficiency was induced in mice using a low lithium diet, and these mice were prone to Alzheimer's, a special species that's been bred for that. So here's the first key finding. By lowering the amount of lithium in the brain through this low lithium diet in these mice, we see the creation of almost 3.5 going on 4 times the increase, 4 times the amount of plaque in those mice who were thus lithium deprived compared to the control mice. A dramatic increase in amyloid plaque. And here, likewise, the low lithium diet of lithium deficiency has created a dramatic increase in phosphorylated tau. This goes on, a couple more slides on this. What you see here is a decreased microglial uptake of amyloid. The microglia are the cells responsible for getting that amyloid out of there and clearing it. So we see then a decrease in amyloid clearance overall as well. So to summarize just this far, the low lithium diet seems to accelerate Alzheimer's pathology, more plaque, more phosphorylated tau. So the implication then is that lithium prevents plaques and tangles, the tangles that come from that phosphorylated tau protein. Well, that's the key idea then, right? Is can lithium do this? Well, we'll see more data on that in just a moment. But this is the development of that hypothesis and the initial proof thereof. So there are some associated results that come that we can see in these mice who are made lithium deficient. And then we'll look at using lithium to actually try to prevent the development of these plaques and tangles. So these associated findings, first, decreased synaptic connections. You'd expect to see this if neurons are shrinking and dying, that they'd have fewer connections to other neurons. And those little black bumps that you see in the control slide, you see fewer of them in the lithium deficient mice, and you can see the same thing quantitatively in the graph. Because there are fewer synapses, there are fewer synaptic proteins. And myelin, which coats the extensions, the axons of most neurons in the brain, also decreased. If you're familiar with the notion of a transcriptome, you might recognize this graph as showing that there is a microglial shift toward an activated transcriptome, meaning they're involved in an inflammatory process, including the increase in inflammatory cytokines. And overall, the key one here then is mice were not as smart. They have several tests of a mouse's cognitive function that we won't try to get into here. But what you see is decreases across the board in that ability to do cognitive tasks among the mice. but not in their visual abilities or their motor or exploratory interests. So this isn't an across the board wiping out of neurons. It's just getting at the ones that are associated with cognitive function, very much like in Alzheimer's. So what we see here then is that lithium deficiency leads to an inflammatory state, to neuron shrinkage, and cognitive decline. Now the key stuff then, can lithium prevent this process? Well, we're going to look at two different kinds of lithium. Lithium carbonate, which is the typical prescription lithium that we in psychiatry have used for people with bipolar for many years. And then this other stuff called lithium orotate. If you look at the many different forms of lithium that you put in a pill, for example, lithium carbonate and lithium orotate are at opposite ends of this spectrum. The spectrum has to do with conductivity, which has to do with ionization. And the hypothesis was that the other end of the ionization scale, the orotate end, would actually make it work better in terms of getting into getting into this brain process of preventing Alzheimer's. So the experiments, well, you'll see, include both orotate and carbonate. And remember, what we're trying to do here is establish, does lithium prevent plaque and phosphorylated tau? So the mechanism of the experiment, then, you take these Alzheimer's prone mice and you give them either just plain water for months as they age, or lithium orotate in a solution in their water, or lithium carbonate. And we're going to look at mice who were given this either water or orotate or carbonate lithium from middle age, 7 to 9 months of age, that's a middle-aged mouse, to 18 to 22 months. And then the experimenters examined their brains when they'd reached an aged state. And here's what we see. This is the beginning of this crucial experiment. This is the amount of plaque burden when they were just given water over those many months from middle age to old age. And in the diagram, in the micrograph, you can see that's plaque burden, the green stuff. Well, and this is the absolute key result. At these three different concentrations of lithium orotate, far less plaque. Whether you look at the pictures or you look at the graph, it's pretty obvious. Whereas with lithium carbonate, it did not work as well. In fact, it sort of worked, but not very much. As you can see that both in the micrographs and in the graph. Now, well, wait a minute, couldn't that have been the orotate that was doing the good work here? How do we know it was lithium? Well, to show that, the experimenters added one more condition, and that was mice receiving sodium orotate. So they get the orotate, but not the lithium. And as you can see, that didn't work. It was left the mice in the same place as if they had had water. The very same, so our conclusion here then is that lithium orotate prevents plaque and does so better than lithium carbonate. We see the same thing here with phosphorylated tau. You can see the same pattern in the diagrams and in the graph. So our conclusion, and this is the key conclusion before we go on to think about could humans do this, is that lithium orotate can prevent the development of plaques and tangles from the phosphorylated tau. Now that's in mice. So it just takes a small extension then to think that lithium in humans in the lithium orotate form could prevent Alzheimer's. Now, there are going to be skeptics who say, well, this was in mice. We need to see this happen in humans. But that would be mounting a whole new set of experiments, and humans take longer to age than mice. So in order to show this in humans, you'd need trials that went on for at least probably something like 2 years. In fact, there are some such trials I'll show you a little later using lithium carbonate. So we have those trials with lithium carbonate, but it'll be quite a while before we have such trials with lithium orotate. And besides, lithium orotate is not trademarkable, not patentable. You can buy it in a grocery store now. So who's going to pay for such a study that will add to the delay before we see that kind of work? So I would say to the skeptics, you want people to wait. I think I'll make a whole separate video about that. In the meantime, there's one other facet of this story that I think is really important, especially for psychiatry, and that is that lithium orotate lowers an enzyme called GSK3 beta, and it does so better than lithium carbonate. Okay, it's one more element in this story of that lithium can indeed prevent the development of plaques and phosphorylated tau. But this is particularly important in psychiatry because GSK3 beta has already been recognized as a key enzyme in the development of bipolar symptoms. So imagine what this means for psychiatry. If lithium orotate works better than lithium carbonate, at least here in mice, at least in terms of targeting the development of Alzheimer's kind of condition, I think we're going to have to rethink the whole use of lithium in psychiatry. And that would be particularly important if lithium orotate was less risky, less toxic than lithium carbonate. I'll come back to that notion of orotate safety in a little bit. But first, what we just saw was like middle-aged start of lithium orotate. What if it didn't get started until later in life? Can it still work? Well, so for this set of experiments, the investigators looked at what they call adult mice, 6 months old, versus aged mice who were 24 months old and had never received any lithium, versus aged and who had received lithium orotate. The aged LIO group, they were 24 months old and had lithium from 12 months, which is kind of like after middle age in a mouse. And what you see here is that compared to the aged mouse without lithium, the aged mouse with lithium orotate looks rather like the younger mouse, the 6-month-old, here in terms of the clearance of the amyloid protein. And the same result here, the aged with lithium mice, shown in the green boxes, are looking more like the control mice, the adult 6-month-olds, here in terms of preserving synaptic spines. And then another key result, an older mouse, kind of like me, is slower to learn a new thing. So the latency before they get it is longer, as you can see with the green and the blue dots. But compared to the gray dot of the 6-month-old mouse. So they're slower, but look what happens. As they get trained a couple days in a row on how to do a task, the aged with lithium mice can learn, and they learn at the same rate as the 6-month-old mouse. So learning new stuff, learning where I left my keys, is actually the same, very much more like a young mouse in that lithium preserved learning ability compared to the aging process in an Alzheimer's prone mouse without lithium. So, in Alzheimer's, people, their memory for distant past is pretty good. Often they can remember where they grew up and where their friends were, but they can't remember where they put their keys, or when it gets really bad, they can't remember who you are. So it's that modern learning that fails. And this shows the preservation of modern learning in these mice. So our conclusion here, lithium orotate can prevent the low lithium consequences, including cognitive decline, even if you started a little late. Just 2 more sections to go here. A relatively short one here on the risks of lithium orotate. Well, the risks from lithium carbonate that we know very well in psychiatry have to do with kidney function and thyroid function. But for lithium orotate, we don't have any data in humans yet, despite the fact that humans have been using lithium orotate, thousands of them for years. As I've heard on my old website, people used to write in about my lithium orotate page. That was 10, 15 years ago. A lot they wrote in. So a lot of people have been doing this for a long time. You would think that if there was a big problem with this, we would have discovered it by now, but we don't have systematic data on this. So certainly the skeptics will say, we don't have the data we need to establish the safety of lithium orotate yet. Well, in the mice, what you see in this graph is that their kidney function, if they got lithium orotate relative to lithium carbonate, no problem. The dose of lithium carbonate was low enough to not bother these mouses, mice, the kidneys of these mice. Now, here's a little bit of an aside, but you'll see why this is important. There's an article from just a couple of years ago showing that lithium orotate is less toxic than lithium carbonate. Well, okay, cool. That's the same as what we're seeing here. So this experiment from the Nature group is a replication of that study from 2 years ago. Well, why is that important? Because way back in 1979, there was an article by a very respected bipolar researcher, Morgan Shao, that said, oh, look, lithium carbonate is less risky than lithium orotate. Lithium orotate caused more kidney problems in mice. But ever since then, people have been waving their hands saying, that wasn't fair. You used the same dose of orotate as the dose of lithium carbonate, but lithium orotate is more effective at getting lithium into cells, so you need to use much less lithium orotate for a fair comparison. And that 2023 article in the upper right, that is the fair comparison. And lo and behold, they did indeed see that lithium orotate was even less toxic than lithium carbonate. So that's why this new study is a really important replication of the 2023 study, so that now psychiatry can begin to go, okay, well, wait a minute. We have two very detailed, very rigorous studies showing that lithium orotate has less risk in mice than lithium carbonate. We need to start investigating that in humans, and maybe we need to start using lithium orotate instead of lithium carbonate in humans. There are a couple studies about just that underway now, fortunately. So that data we will have sooner. In the mice, thyroid function was unchanged, whether they got the orotate or water as they were growing up. But in humans, that might be a little different. I'll tell you just a little bit about that as we get into dosing, just briefly. So in conclusion on risk, it looks like it's low. In mice, it was basically zero as far as kidney and thyroid function, but we need data, and I think especially on thyroid in humans. So now, what dose? We leave now the Nature paper at this point, and this is just my take on, if you have all this information, what are you going to do with it? What dose, if any, of lithium orotate might one consider taking based on all this information? Well, the answer is nobody knows, because this hasn't been studied in humans for the purpose of preventing Alzheimer's. No systematic study. I suppose you could say the younger you are, the lower the dose, because when you get older and you're closer to developing Alzheimer's, you might want to take a higher dose to hedge your bets if you didn't know what dose to be used. But that's just conjecture as well. So let me give you a way of thinking about this. I started by trying to calculate, well, how much lithium actually did the mice get? And my math's so bad, I'm not confident in my equation. And actually, it's simpler than that, I think, as I hope now to show you. But just take me to the end of this video, just a few more slides, hang on. So in Denmark, there's lithium in the water, the natural water supply, and it's higher in those darker green areas. So a person living in Denmark might be exposed to lithium all their lives, drinking their water up to 30 micrograms per liter. And the importance here is that in those areas of greater lithium exposure, the darker green ones, there were slightly lower rates of the developing Alzheimer's. Very important paper, a big hint that this whole line of thought is correct. But the problem is that maximum of 30 micrograms per liter is not very high. It's not that different than zero. For example, in Japan, from a different set of studies, there's twice as much in some parts of Japan, 60 micrograms per liter. Well, in Texas, there are some places where there's nearly 10 times that um, uh, in the northwest part of Texas. And in the Atacama region of Argentina, where lithium is mined, um, there are Andean villages there with as much as 1,000 micrograms per liter of lithium in their water supply. This article was about thyroid. It was observed that some women in these villages had some hint that they were moving in the direction of hypothyroidism as lithium can cause. So I think we still need to think about thyroid when we're up into this range in humans. Now, lithium orotate, the smallest pill that they make that I can find is a 5 milligram. That would get you 5 times as much as Argentina um, in one pill per day. Perhaps if the Andean women were drinking 5 liters of water per day, as they might in the desert, this would be in the same ballpark as Andean water in that part of the Andes. So I think we might say that at least for now, the simplest way to do lithium orotate is this 5 milligram pill. It's probably in a rough, really rough right ballpark. Maybe more would be better. We need some more data to really actually know that in humans. For comparison, then, a single pill of lithium carbonate, the smallest one that's made, 150 milligrams, has way, way more lithium in it. But that's lithium carbonate. And remember, we just saw lithium carbonate doesn't work so well. Um, so that's just for a comparison to show you that the lithium dose that we're talking about in lithium orotate is much smaller than even the smallest lithium carbonate. Now, there was a randomized trial of using lithium for the prevention of dementia that was published back in 2019. I've reviewed that elsewhere, but you might go, wait a minute, how come you didn't tell me about that? Because that did show that lithium seemed to be associated with a reduction in the rate of developing Alzheimer's. But it was a small trial that's kind of been dismissed, even though the result is so important. But look at the doses that they were using there. They started with the 150 milligram lithium carbonate and then went up to 2 or 3 pills a day. So the doses were getting well beyond what we might think of in terms of lithium orotate. And yet they had very few people old. They were all old people in this study, and very few people had problems in terms of kidney or thyroid, for example. So in that respect, lithium carbonate, which might not be as safe, was safe at these doses. Again, suggesting that a tiny bit of lithium orotate, that I think when one thinks in terms of safety, it's looking better than lithium carbonate at these doses. So for now, well, what if you were older? Or what if you knew someone who had minimal cognitive impairment and you were thinking about this as an intervention we need to do now? 20 milligrams of lithium orotate, no one knows the answer to that. Again, for comparison, there is a study that's just wrapping up now. In fact, the results have been submitted. And look at the doses that they used here, a much higher 600 to 900 milligrams of lithium carbonate. And so much bigger exposure. That trial is said to be positive in the sense that it too showed a reduction in the development of Alzheimer's in the group that was randomized to receive lithium. Very important result. We should have that result out anytime, like within the next week or so, I hope. But suffice to say that bigger doses might be relevant in people who already have minimal cognitive impairment. So, and one last thought. Well, those two different versions of lithium orotate, 20 milligrams, one costs $15, one costs $50. So buyer beware. And then you have to wonder, well, how much lithium orotate is really in these things? Is anybody watching? And that's the problem when we move into over-the-counter lithium orotate is it's not like the prescription lithium which isn't necessarily perfect, but it's far more likely to have in it what it says it has. That's not an endorsement for prescription lithium. It's just a caution about lithium orotate. So in summary, these are the three main findings. Lithium has a key role in the prevention of Alzheimer's. That notion is very much strongly confirmed by these data from the Nature publication. It looks like a tiny dose of lithium orotate could indeed delay dementia. We certainly want data in humans, but some people are wondering what to do in the meantime. That could be a long meantime. Um, and lithium orotate is likely better for this than lithium carbonate, at least in terms of what we know from mice. A quick disclosure, I hope you noticed I haven't tried to sell you anything, but I will acknowledge that that is my hand and my 5 milligrams of lithium orotate. I believe in this, and so yes, I am taking it. It could perhaps make me a little too strong in trying to sell it to you, the idea. You can factor that in. When I complete other presentations, like on the randomized trial that's about to come out, we hope, I'll link them here. Otherwise, you can go back to the table of contents for this psych education YouTube channel. And thank you very much for watching.